The human body is made up of tiny building blocks called cells. These cells have a central part called the nucleus. Inside the nucleus, there is a special substance called DNA that holds all the information. DNA, an essential molecule, intricately governs and influences a myriad of traits and characteristics, encompassing not only physical attributes like body color and size, but also a multitude of other biological aspects. By skillfully leveraging and optimizing the capabilities of these remarkable information storage organelles, we have the extraordinary opportunity to catalyze a transformative revolution within our medical sector. In this video, I will explore the vast range of applications of biotechnology in the field of medicine, focusing specifically on its role in diagnosing diseases and disorders. Biotechnology has made significant contributions to the medical sector in disease and disorder identification as well as prevention, particularly in the context of genetic disorders. The application of biotechnology in medical sector is categorized into four main categories. The first and second categories involve the diagnosis of specific diseases and genetic disorders, while the third and fourth categories focus on the prevention of diseases and genetic disorders. The first category is diagnosis of disease. The term disease is commonly used to describe conditions caused by infections, as they involve an external pathogenic organism like viruses, bacteria, and others that disrupts the normal functioning of the body and leads to specific symptoms or physiological changes. In this process, this pathogenic organism is diagnosed. Molecular diagnosis and immunological diagnosis are most useful methods in this process. Molecular diagnosis methods involve using techniques to analyze specific molecules, such as DNA or RNA, for the purpose of diagnosing diseases or disorders. And there are several steps in molecular diagnosis methods. The first step is sample collection. During this step, biological samples such as blood, saliva, tissue, or swabs are collected from the patient. The specific type of sample collected depends on the suspected disease and the molecular diagnostic test being performed. The second step is sample preparation. In this stage, relevant biomolecules such as DNA or RNA are extracted from the collected samples. Various extraction methods are available, depending on the sample type and the desired purity and quantity of the nucleic acids to be extracted. The third step is nucleic acid amplification. The extracted DNA or RNA is amplified using techniques such as polymerase chain reaction, PCR, or isothermal amplification methods. This amplification step allows for the specific target sequences of interest to be replicated, making them easier to detect. The fourth step is targeting detection. Various procedures can be employed to detect the amplified target sequences. These methods include gel electrophoresis, hybridization-based techniques, real-time PCR, and others. The fifth step is data analysis and interpretation. During this stage, the data obtained from the molecular diagnostic tests are analyzed and interpreted. The results are compared with known genetic markers, reference databases, or established diagnostic criteria. Bioinformatics tools and software are often utilized to analyze the data and identify disease-related genetic variations or patterns. Immunological diagnosis methods utilize techniques for detecting and analyzing components of the immune system, such as antibodies or antigens, to diagnose diseases or conditions. These methods involve a series of steps in the diagnostic process. The first step is sample collection and preparation. In this initial stage, the appropriate biological sample, such as blood, is collected and processed. The sample may undergo centrifugation or other techniques to separate and isolate the desired components, such as blood serum. The prepared sample is then added to the microplate wells and incubated, allowing the target antigen to bind to the surface of the microplate. The second step is antigen or antibody binding. In this step, any remaining unoccupied binding sites on the microplate are blocked using a blocking agent, such as bovine serum albumin, BSA, or non-fat dry milk. After blocking, an enzyme-linked antibody is added to the microplate wells and incubated. These antibodies specifically bind to the target antigen in the sample, forming specific antigen-antibody complexes. If the antigen is not present, the antibodies will not bind. Third step is washing to remove unbound components. During this step, a washing buffer containing a mild detergent, such as Tween 20 or Triton X100, is added to the microplate wells. The washing buffer rinses the wells, removing unbound or non-specifically bound components. This step reduces background noise and enhances the assay's specificity by eliminating unwanted substances. Thorough washing ensures accurate detection of the target antigen-antibody complexes. The fourth step is detection and signal generation. After washing away unbound components, a detection reagent, such as TMB, tetramethylbenzidine, DAB, 3,3-foot-diaminobenzidine, or other suitable substrates, is added to the sample. The detection reagent interacts with the enzyme-linked antibody and antigen-antibody complexes. This interaction leads to the development of a detectable signal, such as a color change or fluorescence. The intensity of the signal is proportional to the amount of antigen. Last step is signal measurement and data analysis. In this step, the resulting color signal is measured and analyzed using specialized equipment or techniques. The intensity of the color signal is proportional to the presence and quantity of the target antigen in the sample. The data obtained is then subjected to statistical analysis and compared to known standards or controls to determine the presence and level of the target antigen. The second category is diagnosis of genetic disorder. Genetic disorder refers to a condition that is caused by abnormalities or mutations in an individual's genes or chromosomes. These abnormalities can affect the normal functioning of the body, leading to a wide range of symptoms and health problems. The diagnosis of genetic disorders involves several steps. The first step is a clinical assessment of the patient. In this step, information is gathered about the patient's medical history, conducting a physical examination, and evaluating the patient's symptoms. The healthcare provider collects relevant data to guide the diagnostic process. The second step is genetic testing. Various genetic testing methods can be employed depending on the suspected genetic disorder, such as chromosomal analysis and molecular genetic testing, etc. Chromosomal analysis techniques are used to examine the structure and number of chromosomes in a person's cells. These techniques help identify chromosomal abnormalities that may be associated with genetic disorders. Molecular techniques, including polymerase chain reaction, PCR, DNA sequencing, or targeted mutation analysis, can be used to identify specific genetic mutations or variations associated with the disorder. These tests focus on specific genes or regions of the genome known to be involved in the disorder. The last step is data analysis and interpretation. In this step, genetic testing data are analyzed and interpreted. This involves comparing the results with known reference sequences, established diagnostic criteria, or mutation databases. Like, share, and subscribe to this channel.